Good day, Year 9, and welcome back to Science, where today, as you can see, we're going to be looking at the process of osmosis. So last lesson was diffusion, which is the movement of particles, one of the ways in which particles can move. Osmosis is a special type of diffusion. So that is what we are going to be focusing on today. Before we start, I've got an exam question that I would like you to have a go at. As you can see, key terms are highlighted in blue. The general kind of topic is in green, and the command word for the question is in red. So the command word for this one is describe. So diffusion is an important process in animals and plants. The movement of many substances into and out of cell occurs by diffusion. Describe why diffusion is important to animals and plants. And we should be referring to the different places where it happens in animals and plants. And what is actually happening. It's going to be worth 6 marks. So this is a good time to practice some of those 6 markers. So I'd like you to pause the video and have a go at doing a 6-mark answer for this question. Okay, welcome back. Hopefully we have had a go at that 6 marker. I'm going to put a mark scheme up in a second, so you'll be able to have a look at your answer, and if you could tick where you have done certain things to see how you have gotten on. If you do not feel as though you did as well as you could have done, then please make sure that you go back and have a look at the lesson of diffusion, which was last lesson. What do we need to be talking about? On the left-hand side, you can see what the examiner will actually have when they are marking your GCSE papers. It's not as simple as one piece of scientific information is 1 mark. What you actually end up having is being put into these different levels. So level 1, if you achieve that, will be worth 1 or 2 marks, depending on how much of it you do. Level 2 will be worth 3 or 4 marks, again, depending on how much of it you do. And level 3 will be worth 5 to 6 marks, again, depending on how much you have done. So you can see for level 1, you need to give an example of diffusion, giving a named substance, or you could give a process of where it is happening, or the idea of why diffusion is important. For level 2, you need at least one example and link to a process in either animals or plants. And to get the top level, level 3, you've got to have a description of the process occurring in either animals or plants that is correctly linked to a substance. So what is it that is diffusing? And a process occurring in the other type of organism that is correctly linked to a substance. So you need to be talking about 1 for animals and 1 for plants, an example, and what it is that is actually moving. On the right-hand side, you can see that there are some examples of the things that you could be talking about. So in the first bit, you can see it talks about why diffusion is actually important. Excuse me. So you've got substances moving out that are used by the cell, like respiration's going to use oxygen, things like that. And underneath it, you've got some of the examples that you could be using. So you could be talking about gas exchange, so O2 going into the blood, carbon dioxide diffusing out. Or you could be talking about gas exchange in plants for photosynthesis. Or you can be talking about the absorption of molecules like glucose for indigestion. You could be talking about water absorption in large intestine. Now, we haven't mentioned that. That comes up in a later chapter when we do digestion. You could talk about water being lost through the leaves or transpiration. You could talk about water being absorbed by roots. So both of those came up in previous lessons. And you can talk about minerals also being taken in by the roots. So have a go at deciding how many marks you think your answer is worth based on that examiner's mark scheme. Okay, so we're going to be looking at osmosis today, and what do we need to be able to leave this knowing? So we need to be able to describe what osmosis is. So we need to know the definition, how it works, what it is that it involves. We need to be able to compare it to diffusion. So we need to be able to look at similarities and differences between osmosis and diffusion. And you might be able to explain how different models show osmosis working, or talking about terms that we're going to cover such as isotonic, hypertonic and hypertonic. Okay, now from those words that I just used that will probably be new to you, I want you to think about where you've heard hyper, hypo and iso before. So they are used as prefixes, so before words. So can you think of any examples of where you've heard those terms being used and what it actually means? So have a think about it. You can pause the video if you want. Okay, so for your notes, what do we need to know? We do need to know these three terms, okay? So iso, hyper, and hypo. We're going to be using it in the next slide in reference to tonics. So that is not the drink tonics. That is looking at different solutions, and it's a way of describing the concentrations of solutes that are dissolved in them. So iso means equal to, so isometric, and what we're going to be referring to in the red, isotonic, which means that the concentrations will be the same. Hyper means high, okay? So you might have come across the words hyperactive or hyperthermia, as opposed to hypothermia, which means for hyperactive means you've got very high energy. Hyperthermia means that you're too hot. And so when we refer to hypertonic, it means that the solute concentration is higher. Hypo means low. Okay, so hypoallergenic means lower allergens present, so you can get hypoallergenic dogs. That just means they molt less, produce less dander, which causes allergic reactions. You should have heard about hypothermia, so that's when you get really, really cold. And so hypotonic just means a low concentration. So here's for your notes. So if you could pop this into your book. So hypertonic means that there is a higher solute concentration. So this, we're going to be talking about cells in solutions. So if the solute concentration is higher, then it would be hypertonic, whichever one is the higher solute concentration. Hypotonic means lower solute concentration, and isotonic means that the solute concentration is the same both inside and outside the cell. So if you could pop those down, you may wish to pause the video to make sure that you have enough time to get that into your notes. Okay, so what we're going to do now is I have put onto the slide a link to a video about osmosis. Okay, so what I would like you to do is pop down the 3 questions or 3 things that you're going to be taking notes on, which is what is osmosis, why is it a passive process, and what passive means, and the direction of the concentration gradient. So is it going from high concentration to low concentration, or is it going from low concentration to high concentration? I will be attaching this link into Show My Homework. that is easier. If you know another way of doing it, then by all means, please do. But I'd like you to watch that YouTube video and then make notes on those three things. Okay, so if you could pause the PowerPoint or exit the PowerPoint so that you can do that task. Right, so I hope you enjoyed the video. It's a bit of fun. Can you put a definition, unless you already have it in your notes, a definition for what osmosis is? And this is going to be worth 3 marks, so we need to have 3 main points to it. Can you also put down what a partially permeable membrane is? So again, 2 marks. So we need to be thinking about 2 things, or perhaps for the second mark, you could give an example of a partially permeable membrane. If you would like an extra challenge, can you have a go at putting down what the differences are between osmosis and what we discussed in last lesson on diffusion? So if you could pause this video now, please, and answer those questions. Okay, welcome back. Hopefully we've had a go at those questions. So what osmosis is, what a partially permeable membrane is, and what the differences are between osmosis and diffusion. So let's have a look at the ideal answers. Okay, osmosis, it's the net movement, so the overall movement of water molecules, and we must make sure that we are being specific to water molecules, from an area of higher concentration of water, or dilute solution, so less solute, to an area of lower concentration of water, or a concentrated solution, which means that there's a high amount of solute dissolved in it. And it happens through a partially permeable membrane. So in order to get those 3 marks, we should have mentioned about the water molecules. The bells are still going, so I do apologise for that. We need to be talking about the water molecules. We need to be talking about the different concentrations, so that it goes from high concentration of water to low concentration of water. And we need to say that it happens through a partially permeable membrane. So what is a partially permeable membrane? For the second question, it's a membrane that has tiny holes in it which allow some molecules to pass through. If you said that it only allows small molecules to move through, that's absolutely fine. An example of it, a very important one, is the cell membranes. So that layer that surrounds all cells, be it plant and animal eukaryotic cells, or bacterial cells, which are prokaryotic. Now you might have a go, hopefully did have a go at the similarities and differences between them. So how osmosis and diffusion are similar is that both of them involve the movement of particles from high to low concentration, and neither of them require energy. So we say that the process is passive, as opposed to, in a couple of lessons, we're going to do active transport that does require energy. How they're different is that osmosis will always involve a partially permeable membrane. In diffusion, it can happen across a membrane, but it's not essential. It's not always have to be there. And in osmosis, it's talking about water particles specifically, whereas diffusion is a more general idea of particles. Okay, so we mentioned about hyper and hypo and isotonic solutions, and this is probably the hardest thing that students struggle with in terms of getting your head around it. Now, if there is a high solute concentration, that means there'll be a lower water concentration. So if I got a glass of water, which would be the solvent, and I put some salt in it, the salt would be the solute. So if I had a glass of water and I put in 6 spoonfuls of salt and stirred it up and dissolved it in, that's going to have a high solute concentration compared to if I had a glass of water where I'd only put 3 spoonfuls of salt in. That would have a lower solute concentration. Now, if something has a lower solute concentration, it means it's going to have more water. So another way that you can think about this is if you drink squash, orange squash. I'm quite partial to Tesco's cherries and berries squash. If you put in more squash into your glass, then you're going to end up with less water in there. So that will be a higher concentration. You might even say you like it concentrated. Whereas a dilute one, or people who don't like a strong taste, you'll only put a small amount of squash in there and lots of water. So that's got a lower solute concentration, lower squash concentration. So in the picture, we've got 3 different examples. There is a dotted line between the left and the right-hand side, and that is the partially permeable membrane. Now, you'll see on this diagram that it's labelled as a semi-permeable membrane. Be aware that you might see it as either semi-permeable or partially permeable. It means the same thing. Now, if we look at the first diagram, you will see that on the left-hand side, there's only 2 red circles. Now, those red circles are representing particles. So because it has 2 red circles and lots of water, that means that it's got a low solute concentration. So you can see that it's been labelled as hypotonic. On the right-hand side of that first diagram, you've got 4 red particles and less water. So that's much more concentrated. So we refer to it as hypertonic. Now, if we look at the middle one, you can see that both sides are isotonic. And you might be thinking to yourself, well, there's 4 red particles on the right-hand side and only 2 on the left. So it can't be the same, but it is. Because you can see that there is less water on the left-hand side than there is on the right-hand side. So in relation to the particles, concentration in relation to the water, they actually have the same concentration. On the diagram on the right-hand side, you can see that it's very, very similar to the first one, but it's just the other way around. So you've got a hypertonic side on the left because there's more particles, less water, compared to the right-hand side, which has more water and fewer particles. Now, on each one, you will notice that there is an arrow, and that is showing the direction that the water particles will move. So it's going to be going from a hypotonic to a hypertonic. So it's going from the low solute concentration, which means it's got high water concentration, to the high solute concentration, which means it's got low water. Okay, so it's always going from high water to low water. So the one on the left is going from the left-hand side to the right-hand side. The one in the middle is going both ways at an equal amount. And on the right-hand side, it's going from the right to the left. Now, in the text below, you will see that it refers to something called water potential. Now, this you don't need to worry too much about, so you don't need to make a note of water potential. But it's basically for something, lower water concentration means lower water potential. It's a term that will tend to be used more later on and at A level. Now, depending on whether you put cells in a hypertonic solution or a hypotonic solution, will determine what happens to that cell. And plant and animal cells will do things differently. Now, in the next lesson, we're going to be focusing on osmosis in plant cells, which goes with a required practical that Miss Corbett will be recording, which is looking at osmosis in potato cells, which will be the plant cells. This lesson will mostly be focusing on osmosis in animal cells. So osmosis in plants and animal cells is different because they've got those different structures. You might be able to think about what that structure is that is going to have that effect on water moving in and out of the cell. Plant cells can take on a lot of water because they have a cell wall, which is that structure I was referring to. So they don't burst. In animal cells, if you put in too much water, they do burst. So imagine blowing up a balloon. The balloon on its own would just be the animal cell. If you keep blowing up the balloon, it's going to burst. But if you put that balloon in an empty water bottle and started blowing it up, it would reach the volume of the water bottle and it would stop. And that water bottle is acting like the cell wall. Now, if animal cells lose water, so if you put it in a solution where water is moving out of the cell, then they shrink. Okay? Seems straightforward. And if you put plant cells in them, they don't shrink, they become plasmolysed. Okay. Now, what plasmolysed means is that the vacuole pulls away from the cell membrane. So it doesn't shrink the overall cell because it's got that cell wall still on the outside, but it's that vacuole moving away from the cell membrane. That's what plasmolysed means. And we're going to be coming across a few terms that we need to know for animal and plant cells. Okay, so if you have a look at the diagram on the right-hand side, you will see that there are some blood cells, some red blood cells. Now, these are animal cells, and in the diagrams, it's got two different scenarios, one where water is moving into the red blood cells and one where it's leaving. So if we look at the diagram, when cells are placed in distilled water, so it's pretty much pure water, water will move into the cell because it's going from the high water concentration of the... Oh, that's another bell. Sorry about that. Because it's moving into the cell, because it's going from that pretty much pure water concentration, high water concentration, into the lower water concentration that will be inside the cell. And that causes the cell to swell and then will eventually burst. And that bursting of the cells is called lysis. So lysis is when animal cells take on too much water and they burst. If we follow it through on the right-hand side, these are red blood cells that have been put into a concentrated salt solution. So there is a high solute concentration, very low water. So water leaves the red blood cells and goes into that salt solution, and it causes those cells to shrink and shrivel. And obviously that means that those red blood cells won't be able to function properly, and we call this crenation. So they have been crenated. There is a link to a very exciting graph at the bottom of this that you may want to have a look at, that will go over some of these processes. So what I'd like you to do now, on the left hand side you can see an animation, or it's looking through a microscope at some animal cells that have been put into a solution that has a very, very high water concentration. Okay, so what I would like you to do is have a go at describing what is happening to those cells in terms of osmosis. So we need to be talking about the difference in the solute concentrations. So is the solute concentration higher inside the cell or outside the cell? We need to talk about the movement of that water through the partially permeable membrane. We need to mention the words hypertonic or hypotonic or isotonic, depending on which one you think it is, and what effect that is having on those animal cells. So if you'd like to pause the video and have a go at putting that into your own words. Okay, hopefully we've had a go at putting that into our own words. If you could check that you have included the information that I'm going to talk about in your descriptions, that will be great. So hopefully you have noted or said that the solution that the cells are in is hypotonic, which means it will have a low solute concentration and a high water concentration. This means that the water particles will move from the solution into the cell through the cell membrane, which is a partially permeable membrane. Because the cell is taking in all of that water, is causing it to burst, and that is lysis. So hopefully we've got that. Hopefully we've figured out that it's in a hypotonic solution. So the water goes into the cells and causes that lysis. Okay, so we're going to do some examples now, and you've got to decide which way the water particles are going to go. So for this first one, the red circle is representing a red blood cell, and in the red blood cell there is a low concentration of water. If I put that cell in a solution that has a high concentration of water, which way is the water going to go? Right, hopefully we guessed, well, didn't guess, we realised that the water is going to move into the cell. So it's going to go from that high concentration of water in the solution into the cell, which has a lower concentration. What will happen to the cell as a result? Think about that answer. Okay, hopefully you have said that it is going to burst, which is lysis. Okay, next one. So in this example, we have got an equal concentration of water. So this is isotonic. The concentration of the solution is the same as that inside the cell. So which way is the water going to move? Realise that the blue arrow has already given it away. It's going to go in and out an equal amount. That almost rhymed. So there is no net movement. So some is going to move in, some is going to move out, but the overall movement is 0. Okay, so in this example, this time we have a high concentration of water inside the red blood cell and a lower concentration of water outside the cell. That lower concentration of water is the solution. So would we describe that solution as hypertonic, hypotonic, or isotonic, do you think? Have a think. Okay, hopefully we guessed that it is hypertonic because it's going to have high solute, which means low water. Right, which way is the water going to move? Is it going to move into the cell or is it going to leave the cell? Hopefully we realise that the water is going to move from the cell into the solution because it's going from a high concentration of water to a low concentration of water in that hypertonic solution. What's going to happen to that cell then? Have a think. Okay, hopefully we said that it's going to shrink or shrivel, both, which is called crenation. Okay, so it's undergoing crenation. Now, I'm sure you will agree with me on the next slide This very interesting example of osmosis. You do not need to write any of the following or make note of any of the following. Is purely to see this happening in action. Okay, so this on the left hand side you can see a scientific diagram of a cell called a paramecium, and on the right hand side you can actually see that one of those paramecium under a microscope. Now, paramecium live in fresh water, so they'll live in streams and lakes and things like that. And because it's in fresh water, there's a very high water concentration in that fresh water. There's not, say, lots of salt as there would be in the sea. So water is constantly moving into this paramecium cell because it's going from that high concentration on the outside to the lower concentration on the inside. The paramecium has not got a cell wall, so it's only got the cell membrane. So it needs to make sure that it's getting rid of all that excess water so that it doesn't burst. So what it's got, which you can see happening in the animation, towards the top of the cell, there's one on the left and one on the right. The two things that look like they're getting bigger and then popping, those are special vacuoles. And what happens is all that excess water goes into those vacuoles, see, increases in size, and then it will fuse with the membrane and let all of that water out of the cell. So it's constantly getting rid of that water that is going into the cell. Like if you're on a sinking boat and you have got a bucket and you are bucketing out all of the water that is coming in, it's constantly doing that. So what we're going to do now is have a go at a couple of 4-mark questions, which is going to be getting you to apply, and one of the questions getting you to apply what we've done to a new situation, which is what's going to happen in the exams. So question number 1 says explain, so we need to have a because in that sentence, explain why it is important for the cells of the human body that the solution concentration of the fluid surrounding the cell is kept as constant as possible. So it's asking you to say why it's important that the concentration around our cells doesn't change. It's always staying at a constant level, the same level. And that's going to be worth 4 marks. That's going to be quite tricky. Maybe think about what will happen if the concentration outside the cell of water goes too high or too low, what effect that has on the cell. Question number 2, animals that live in fresh water have a constant problem with their water balance. The single-celled organism amoeba has a special vacuole in its cell. The vacuole fills with water and then moves to the outside of the cell and bursts. The new vacuole starts forming straight away. Explain in terms of osmosis why the amoeba needs one of these vacuoles. So this is very similar to the last slide where we were talking about the paramecium. The amoeba is a fantastic single-celled organism. You can see an amoeba in action in both of the animations. They're both showing an amoeba. So you've got to talk about why that vacuole is so important in an amoeba which lives in fresh water. So can you have a go at those questions? Please can you pause the video and then we'll go over the answers shortly. Okay, so hopefully we have had a go at those questions. Both of them were 4 markers. One of them is talking about the importance of keeping a constant concentration of water around animal cells and then applying it to an example of the amoeba in fresh water. So we're going to go through, tick where you are getting these marks from. It doesn't need to be the exact wording that is going to come up, but words to that effect. So in question number 1, which was explaining the importance of the cells of the human body when they're in a solution, that it's staying at a constant level. Here are some examples. You notice that in brackets I've put what would be worth 1 mark in an exam. So I'll read through an ideal answer and you can see where it is that you're picking that mark up. So if solute concentration outside body is more dilute, means higher water, than the cell contents, like I said, hypertonic solution, the water will move into the cells by osmosis and they may swell or burst. Now, you don't need to have all of that information to get the mark. You need to be talking about water. If water moves in, then there's going to be lysis, or the cell bursting. If solution concentration outside the body cells is higher than the cell contents, or is a hypertonic solution, the water will leave the cells by osmosis, causing them to shrink and stop working, or crenation. So if you've written in that one, in a hypertonic solution, water will leave the cells by osmosis, causing crenation. Brilliant. Next one, solution concentration outside the body cell must be kept as constant as possible, so isotonic, to minimize changes in the shape and size of the cells, to keep them working normally. Next mark. Because it was an explain question, we need to say why that's going to be important. What effect would it have if they are getting bigger, or lysis, or shrinking for crenation? Right, the next one was applying knowledge to a new situation. So I gave you the example of an amoeba. Okay, now if you have spoken about that the amoeba will be in a high water concentration in the fresh water, or you could have said that it's in a low solute concentration or a hypotonic solution, then that's going to pick you up a mark. If you said about the cell membrane being partially permeable, so water keeps moving in through osmosis, that would be worth 1 mark. If you had said that if the water keeps going in, it will burst, the cell will burst, or the organism will burst, or lysis of the cell, then that would be a mark. And if you said water is moved into the vacuole and then removes the excess water, that would also be a mark. So some quite complicated higher level answers to appreciate, but hopefully you were able to pick up 1 or 2 marks, if not 3 and 4 marks. So this comes to the end of this lesson. So hopefully we have taken from this lesson what osmosis is, the movement of water particles from a high water concentration to a low water concentration to a partially permeable membrane. Hopefully we know what hypotonic, hypertonic and isotonic solutions are. Hopefully we know what happens when we put an animal cell in those 3 different solutions, including those key terms lysis and crenation. So next lesson we will be looking at osmosis in plant cells, which will be a relatively short lesson, but it does come with a required practical that will be videoed by Miss Corporate. So it will probably have on the YouTube channel part 1 and part 2, depending on which you need to do first. Well done for coming on to this and doing the lesson. I hope that you are all staying safe and well and continue the good work. Thank you very much.